This is a production of Cornell University.
Today, I would like to talk to you about exploitation of complex structure of genotype by environment interaction in cassava root crop using IET cassava breeding program as a case study. So multi-environment trials allow breeders to screen for genotypic reaction to a range of environmental conditions, a phenomenon people refer to as a genotype by environment interaction. This kind of trial is usually conducted at uniform yield trial at the late stage of evaluation prior to release of varieties. A number of statistical methods have been proposed for exploiting genotype by environment interactions. So among them are linear by linear models such as additive May effect and multiplicative interaction model, site regression model, factor analytic model, and multivariate multi-environment model. So the principal objectives of this study is to assess complex structure of G by E interaction by identifying optimal model with preferred various covariance structures. So we're also interested in estimating genetic merit of individual genotype toward identification of stable phenotype. So to achieve these objectives, so we try to put together a total number of 30 trials across 9 locations in 3 growing seasons. So those trials were established as a randomized complete block design interior replicate. So in total, we have a total number of 116 genotypes evaluated across 30 trials. So in this study, we intend to make use of R, SAS, and AS-REMO for the analysis. So as you can see from this table, this shows the number of trials for individual environments and across the growing season. And you can see that the data exhibit a kind of unbalanced structures for the fact that the number of trials vary from one location to the other and from one growing season to the other. So to start with, we try to come up with this concurrence metrics to have idea of the number of genotypes that is common to any pair of environment. So the key message here is that those environments that have high number of genotype in common, so there is high likelihood of sharing common information than those environments that have less genotype in common. So after that, we went ahead to have idea of the distribution of the root yield, which is our key trait of interest. So the key message from this box plot is that the yield seems to exhibit a Gaussian distribution from one environment to the other. As you can see that the mean is approximately equal to the median. Another key message from this plot is that there is heterogeneity of genetic variance across the environment, meaning that this suggests the presence of genotype by environment interaction. So as we all know that in a multi-environment trial, the same trait measured in different environments can be considered to be a different trait. So we try to look at Pearson correlations among those environments. As you can see that there are some number of environments where the correlation is very high and positive. So that means there is less of G by E or no G by E in those kind of environments. There are some with positive correlation that are very low, so meaning that there is likely to be G by E in those environments. So we still have some environments that are very contrasting in the sense that the correlation is perfect and negative. So which suggests that there is likely to be presence of G by E, high G by E. So that is a visual approach of assessing the presence of G by E or not. So we went ahead to assess the presence of G by E numerically. So and here we are trying to test hypothesis that there is no G by E against alternative hypothesis that there is G by E, meaning that the variance of G by E is greater than 0. So we fit this nested model for the null hypothesis without inclusion of genotype by environment interaction. So this same model is reparameterized by inclusion of genotype by environment interaction. And this is the result we got from the nested model. And you can see the variance component estimated for each of these effects here. We have the similarity for the full model. So, and these are the various components attributed to each of these factors. So in order to see whether the inclusion of G by E is significant or not, so we have to carry out the likelihood ratio test. And by looking at difference between log likelihood of nested model and full model, so which approximate chi-square distribution with degree of freedom equal to the difference in the number of the parameter estimated. So, and which seem to be significant. So, which tell us that the inclusion of G by E makes sense, and that cannot be ignored. As you can see here from this model, you can see that the variance attributed to G by E is more than twice that of genetic effect, which means that G by E is the genetic variance is specific to environment and is very large, and G by E is very important and cannot be ignored in this study. So we went ahead to see if we should assume that the error variance is similar across all those 30 environments, or are they heterogeneous? So we went ahead by fitting the full model. This time around, we assume that error variance is heterogeneous in each of those environments, and we fit the nested model, which assumes that the residual variance is uniform across all the 30 environments. So this is the result we get by fitting the full model. As you can see, the error variance range from 2.4 to 99.66. And for the nested model, which assume one uniform error variance across all the environment, give us this result. So as usual, we carry out the likelihood ratio test, which confirm that assumption of error variance heterogeneity makes sense, and it has to be taken into consideration so that the result from the analysis will not mislead us. So in the next couple of slides, I'll talk about different kind of various covariance structures we fit in the course of trying to explore the complex structure of G by E. We started by fitting compact symmetry model, which is the most restrictive of all the models. So this kind of model assumes that the genetic variance within the environment is uniform, and the covariance or correlation between any pair of environment is also uniform. So this model may not be realistic, going by what we saw in the previous slide, that the error variance is heterogeneous, the genetic variance within the environment is heterogeneous as well. So this is extract from the model we fitted. So it's 30 by 30 matrices, but there's no way we can visualize everything at once. As you can see along the principal diagonal, those are the genetic variance within the environment which are uniform. Of the diagonal, that is the correlation between any pair of environment. So we went ahead to fit compact symmetry heterogeneous model, which is just extension of the previous model. So here, the correlation between any pair of environment is uniform. And what makes it different from the previous model is that the genetic variance across the environment is heterogeneous. And this is the result obtained from that model. As you can see along the principal diagonal, these are the genetic variance estimated in each of the environment, which is unique, but of the diagonal is the correlation, pairwise correlation between environment, which is uniform. So this is another form of model we fitted, and this is the most liberal of all the model. It is liberal in the sense that it's fit full G by E without structures. So in this kind of model, we assume that each environment has unique genetic variance, and correlation between any pair of environment is also unique. So fitting this kind of model, you have to estimate quite a large number of parameters. Take, for example, with 30 number of trials in our study, so that means we are going to estimate around 465 parameters. So we try to fit it, and this is the error message we got. So this means it is not only computational inefficient to fit this kind of model, but also computational prohibitive. So we fitted a factor analytic model as well. Factor analytic model is nothing but just a mixed model analog of Armin, which is additive math effect and multiplicative interaction model. So this is a model that is a kind of lower order approximation to unstructured models. So with factor analytic model, you can identify the latent common factor that capture and explain the G by E pattern. So as you can see, this is how the structures of the factor analytic models looks like. And this component phi is specific variance in each of the environment, and this square of loadings plus this specific variance will give you the genetic variance in each of the environments. I'm using this to illustrate when you fit factor analytic model of order 1, and of the diagonal is the genetic covariance between any pair of environments, and it's just a matter of dividing this genetic covariance by the square root of the product of this variance and this variance if you are interested in knowing the genetic correlation between any pair of environments. So after fitting all these sort of models, so we try to use goodness of fit statistics to be able to identify the best model that explain the complex pattern in G by E. As you can see from attacking information criterion, so tell us that a factor analytic model of order 2 is the most parsimonious model, explaining the complex structure of G by E with 114 parameters. And by looking at BIC, it identifies compound symmetry as the best model. A lot of people prefer to use AKK information criterion simply because the BIC imposes heavy penalty on the complex model by identifying a simpler model. So we go for AKK information criterion. So after identify factor analytic model of order 2, what I mean by order 2 is that we have two multiplicative components. So, and this is the result we obtain from factor analytic model of order 2. So the first two columns here, that is what we call the latent common factors, which explain the complex pattern of G by E. And this column here, that is what you call specific environment specific variance. In other words, this is the variance in each of these environment that is not captured by these latent common factors. So one good thing about this factor analytic model is that when you have information about environmental covariables like precipitation, the temperatures, so you can decide to look at correlation of those environmental covariables with this latent common factor to be able to identify which of the environmental covariable is very relevant in explaining G by E interaction. So from this factor analytic model, from these latent factors, so when you take the square of this and this plus this one, that will give you the genetic variance in each of these environments. So the key message here is that our factor analytic model of order 2 capture a total of 60.9% of the variations, where the first component and the second component accounted for 42.1 and 18.8% of the variation respectively. So the lesson learned and take-home message from this study is that the compound symmetry model fits a simple G by A structures. They are very easy to fit and convert easily. They are recommended if a better fitting covariance structure cannot be found, even though they do not provide better understanding of G by A structures. Unstructured model is computationally demanding as the number of environments or trials keep increasing. So in analysis of multi-environment trials, it is very rare for people to use this model because you may end up having a solution that is not converged. Factor analytic model is very parsimonious and an effective approach for dissecting G by E in a situation where you have a large number of trials. So the next step is to see how we can identify subsets of environment with and without crossover in these studies. And we also plan to incorporate state marker data into the model to see how we can capture the relationship among those genotypes. So at this junction, I would like to thank Bill Gates Foundation, UK Aid for the financial support. My appreciation also go to my committee member for the pieces of advice, word of encouragement, and thanks to everyone in Joluk Lab and our collaborators as well. I thank everyone for giving me attention. Thank you.
Okay, thank you very much, Moshood. We're running.
This has been a production of Cornell University, on the web at cornell.edu.